The talk's entitled Solutions for Drug Discovery and Development, so we should be finishing on a high. So with that note, yes, it's good to have a challenge, right? Okay, over to Martin, thank you.
So I have a difficult task, of course, because there's not much Theranostics in my talk. But you will see at the end of the talk, with the final slides, I'm coming back to that. While the first part will be more related to what we heard in the keynote session and also in the regenerative medicine session before. So you said it already, so Merck Millipore is a life science division of Merck. So this is a German Merck, KGaA, and we are part of the chemical division. This is Merck Millipore, separated from pharmaceuticals division and Merck Serono there. So we are not a prolonged arm of Merck Serono. We are offering services for drug discovery and development industry as a whole, and Serono is a customer of ours, but not more than a customer. The Merck Millipore division has 3 business fields. So we have process solutions. So these are our colleagues offering workflow solution for larger production, bioproduction of growth hormones, antibody programs, and so this employs pharma and biopharma raw materials, but also specialized filtration technology. So this is this part. Then we have lab solutions. You might have known that Millipore was famous for lab water purification. So this whole part is within lab solutions now. And then we have bioscience, which is providing industry with tools and content, assay contents for life science research. So we have, for instance, flow cytometers there. So we are chief partner of Luminex technology for immunoassays and so on and so forth. So we have the biochem inhibitors, biochemicals within this part, but also a business field, drug discovery and development, and this is what I'm representing, and also Steve Davies, who is in the room here with me from the Dundee side. So, and what is it? What is that business field? So we have lead discovery, as we call it, on the one hand side as a business segment, supporting small molecule lead discovery on major target classes, kinases, phosphatases, GPCRs, and ion channels. And we offer solutions for high content analysis. So I have no slide here, but we have an in-cell analyzer ourselves, and we are supporting customers with kit solutions for toxicity research, whether it's hepatotoxicity, neurotoxicity, or whatever, which can be employed to stem cells as well. And then there is a second division, Biopharma Services. So we call it a niche CRO here, because we are a specialized CRO for large molecule discovery and development. So here we are focusing mainly on peptides, proteins, antibodies. So we are doing bioanalysis, bioanalytics here for biopharma. And all the whole thing together is the discovery and development solution business field. So what are our customers' objectives? So they want to fill their pipeline with potential therapeutics. That's me? No. So they want to identify therapeutic candidates, of course, progress candidates through evaluation quickly and cost effectively, obtain regulatory approvals to market, and of course, we should not forget that contribute to the improvements in quality of life. And we work as a CRO to support them with either isolated services or with more full packages, like for instance, obtain regulatory approvals to market within our business field. So that is our aim. And you can also look at it in a different way. So what we do is, for the early phases, let's say from hit to lead to candidate, preclinical candidate nomination, this is a part of lead discovery we're working, where the data is usually used to guide development of a drug. So what is the right candidate to go forward? Which is a safe, efficacious candidate you want to work with? Whereas the biopharma service solutions are going more after the later phases, so where regulatory agency required testing is necessary, where the data will be submitted to FDA and EMEA afterwards, and where you have to prove that the drug works and is safe. And this employs, as mentioned before, not only the drug but also perhaps exploratory or confirmatory biomarkers that go together with the drug. So we do that on 3 sites. So we have St. Charles in Missouri, which is a former Linko site. You might know Linko, perhaps they had famous immunoassays, radioimmunoassays, for instance. So where we do our cell-based assays for GPCRs and ion channels, but have Biopharma Services as well. We acquired in 2009 a site in Oxford, BioAnalab. So this is a full-blown CRO for bioanalytics for large molecules, founded by Jeff Hale. Jeff Hale is one of the original inventors of the ChemPass antibody, which is a very famous antibody. So they went the whole way from inventing an antibody up to market. So this is a quite experienced team finding solutions for customers to support antibody clinical programs. And then, of course, we have Dundee in further 1 mile, 2 miles up a road. So this is perhaps one of the most famous places, at least as a commercial provider of kinase solutions, including products and services. And Steve, who is here with me, is a managing director of that site. The philosophy for our lead discovery programs is, so we have drug targets, assay reagents, and kit solutions. So what we try to do is to produce the drug targets ourselves. So all the enzymes we are using, or most of the enzymes we are using, are produced by us, as well as the cell lines we are using for GPCRs and ion channels. So that makes us quite independent from third parties, and we can derive services from that for screening, profiling, and custom services. That's what we usually do. It was described before, if you work as a CRO in that field, you need a quite extensive laboratory equipment. Here, for instance, is a subset shown. For instance, on the left-hand side, these are the automated electrophysiology instruments you would use, for instance, for investigating currents in cardiomyocytes as well as in recombinant cells. So these are these automated platforms which are providing the throughput you need to put compounds in a larger throughput through these assays. But we also have, of course, all kinds of plate readers. We have instruments like these MaxCyte machines for bulking up cells and making bulk transfection of cells to support HDS campaigns, flipper machines, and also workflow solutions as well. So it's although we are a smaller part, not Merck Serono, we have quite an impressive set as well. What we focus on primarily in lead discovery is efficacy and toxicity. So these are the major areas where we try to help customers to find a solution. This can be done either on an individual target. This is the main thing we do, but also employed are model cell systems, like, for instance, cardiomyocyte, hepatocytes. So we try to optimize cell systems here as well and bring them into the drug discovery pipeline earlier so that that can be used, for instance, instead of biochemical assays, in the case of kinases, for instance. And what becomes more and more obvious is that you also have to have the right bioinformatics tools to do so. So it's not possible to go with an isolated biochemical assay further. And I have an example here for kinases, simply as we were discussing some oncology approaches here. So if you simply think about the amount of data which is available when you're thinking about a single kinase, or let's say a subset of kinase, might be liability targets of your drug of interest. So you have, in 2010, you had at least 2,400 new papers a month focusing on kinases. You have all kinds of pharma-relevant data, which would be, for instance, in silico docking data, structures that can be heat maps published from somebody, perhaps MBIT data or competitors of ours publishing this kind of stuff. And then, of course, you have all the open source databases supplying you with information. And I think this is mainly unstructured information, and we think one major driver also to help improve decision making will be to structurize this to help customers and scientists to make a better decision how to progress with their compounds by linking in more information about, for instance, toxicology, disease biology, expression, upstream regulation, and so on and so forth. So this is a crucial step. You might think, why is that necessary? I know my target, I know what it does or so. But if you simply look on here, for instance, kinase profiling data, where you have marketed drugs, all of them quite common ones, and also mentioned here before. So, and you plot the kinases screened against them, against the inhibitors, then you see that they have a tendency to be promiscuous, and a scientist has to understand, is that really relevant for my program? Is it something which is desired, which is not desired? So what do I do with all this kind of data? And if you think about a larger organization where they have a couple of kinase programs, so who is doing this? Who is logging this data, whether it's important, for instance, into a database or something? So there are lots of scientists running through the same curation again and again for the kinase programs. And we think we have to find solutions to help customers to get a faster idea of what their compound is doing. And we started this last year with a first attempt. So you have seen the kinase tree before. So this is our version of the kinase tree, but it's not static in a way. So it's an interactive tool where you can plot inhibitor data on the tree and work on it. You can restrict for activity ranges. You can restrict for particular kinase families you want to see, or different mutants, or whatever you want. And what I would like to mention is this part here, not very well shown here on the bottom. So this is at the moment public data information coming with it, including also pathway information and so on and so forth. And this is something we are going to extend in the future to come up with curated literature data, to come up with drug-target interactions, what is known about this target as a liability target and so forth. So I'm not talking about public domain data, I'm talking about curated public domain data, really people, scientists reading through it. And I would like to mention that this version is not only available for kinases, we have it for GPCRs as well. Another trend or a major direction we are focusing on is to employ more physiologically relevant in vitro assays. Earlier in drug discovery, you know, there is a big attrition between preclinical and clinical, and we think this can be avoided in part to bring in more relevant systems earlier in the pipeline. And what we think is, for instance, you can do this with functional assays using native proteins. So the best scenario would be a cell system where you are approaching the, if it's, for an example, a kinase, the native kinase with its native substrate, and you get a measurement for that, so no artificial system involved. You can use therapeutically relevant cells now, like primary cells or embryonic stem cells. You can use high content screening as described in Adam's talk, or something that we call signal profiler, which is going after pathways, bypasses, crosstalks, and so on and so forth. But nevertheless, with this concept to bring up more physiologically relevant assays, or let's say relevant assays, we started to do a safety liability panel with functional screening of 125 targets. So this is a kind of panel each drug compound sees during this development. Most companies do this nowadays, still do this nowadays with ligand binding assays, which give you no idea about whether your compound acts as an agonist, antagonist, as an allosteric modulator, or what it does on the target. And we think that's simply not good enough, and you should get your answer earlier. So we employ functional screening, so flipper screening for GPCRs here, automated electrophysiology for ion channels, so which is comparable to what Adam showed for their cardiomyocytes. So we look on a singular channel level what the compound is doing, whether it's blocking or not. And the targets within these panels are chosen for their relevance to critical diseases and processes. And why you do this, for instance, here is an example for a neuroleptic, clozapine, which is a well-working neuroleptic, and it's known in the literature it's promiscuous, so it works as an antagonist on adrenergic, dopamine, muscarinic receptors, also serotonin receptors. That's well known, but what you can see, it's always working as an antagonist. But there can be other cases, like for instance here, oxymetazoline, it's a nasal decongestant, an agonist for alpha-adrenergic receptors, where you can detect a liability for serotonin receptors, and interestingly, it's not always working as an agonist. There are cases like, for instance, for 5-HT2A, where it can work as an antagonist. And this is something you would like to detect relatively early and have a liability flag around a compound like this, and you should not rely on ligand binding assays, which have also the possibility, the liability or the limitation that they're not the best ones for agonistic programs anyway. Here's another example, somewhat shown like in the cardiomyocyte one. So this is one package we call a cardiac profiler. So we were the first ones to come up with a package like this, where we were screening 8 ion channels in parallel on our cell lines, and what you get is an idea whether your compound has an effect on those ion channels. So it's true, only HERC is requested from the FDA to be tested, but I think nowadays most companies tend to screen more than HERC. So they have their own strategies, of course, how to conduct those kind of studies. In some cases, it's every compound sees 3 channels or 8 channels, depending on what, but every company has a strategy for that. And as I said, so this is automated electrophysiology we are applying for compounds there. The automated electrophysiology is a label-free assay. So you have the cell line, you do not have to put in any reagents to measure it. So therefore, it's a nice approach, as it's relatively close to a physiological system, so you can do it with cardiomyocytes. But of course, this is something you can do only for ion channels at the moment. We were thinking about something which can work as a kinase, cell-based kinase assay, or generally as an assay helping you to highlight cell signaling pathways better. And we came up with the idea to combine Milliplex MAP, so this is our own brand of Luminex assays, together with the Luminex technology and analytes detecting phosphorylation events. So we have roughly 100 at the moment assay kits available detecting phosphorylated status of given substrates, which help you to investigate phosphorylation levels within relevant cell types. You can measure or investigate pathway crosstalks, pathway feedbacks, temporal effects within the cell lysate. And within the cell lysate, you are not limited to artificial cell lines. That can be, of course, tissues, that can be primary cells, stem cells. It's possible to do that. And what you can do, for instance, is you can, with the right mix of analytes, go from the surface of an intermediate to perhaps an endpoint of a pathway, and do this simultaneously while you're using the Luminex system. And here's an example shown, for instance, an early example, because we launched it just recently, where we are following up the EGFR pathway down to STAT3, so with the RAF, MEC1, ERK intermediate there, or the signaling members down to STAT3. And what we simply did was to compare EGFR inhibitors with ERK inhibitors, or with RAF inhibitors in this case, and to see whether we can detect an inhibitory effect of those compounds within the cell lysates, and it's possible. So what you see is that here what we show here is ERK phosphorylation levels. What you can see is that by intervening with EGFR receptor, you can get a readout for ERK1 and 2 for the EGFR inhibitors, but also for compounds interfering with the RAF, it's possible to see this for ERK. So ERK phosphorylation levels went down compared to DMSO, but there are other cases where RAF inhibitors give you an augmented level of ERK phosphorylation. So what we think is you get a much higher information content from assays like this than simply comparing biochemical data. Oops. Now I would like to skip to Biopharma Services, which is perhaps more relevant to Theranostics here. As I already said, so we define ourselves here as a niche CRO as we are focusing on large molecule development, or large molecule bioanalysis. And why is this special? So you might think there's nothing special at all, but I would like to convince you it is. So most companies who are offering, let's say, ADME, PK, DMPK assays and whatever, they are focusing on small compounds. So here in the range of aspirin, ibuprofen or something. We are working with the nasty beasts, which are antibodies, for instance, which are of course much larger, which you cannot detect by using HPLC mass spec technologies. It's impossible. So we are not measuring in most cases the drug itself. We are measuring indirectly with an immunoassay the amount of a drug within a sample. And if you think about an antibody applied or anti-drug antibodies within the body, so we are all challenged every day. We have tons, an ocean of antibodies already in our body, and we want to detect with a bioanalytical assay the one which is given as either a therapeutic or the one which is a particular anti-drug antibody. So this is much more challenging than doing mass spec and, for instance, identifying in a blood sample the amount of a small compound drug. And of course you deal more with matrix problems as well for the immunoassays compared to a mass spec analysis. The Biopharma Service division, so what we have a global footprint. So we have facilities in UK and US, and what we do is we offer pharmacokinetics, toxicokinetics there, immunogenicity assays. We offer support for GMP, so that's potency and stability assays. Within our group, we are not supporting GMP as a manufacturing process. What we do is we help customers with documentation and these assays. So we do the potency assays, stability assays for them, report back the data in a form that it's used with regulators. So it's not that we are doing the manufacturing ourselves. And we are working on biomarker assays. But I would like to say here directly, the biomarker assays we are working on are protein biomarkers only. So we are not engaged in any genetic assessments, nothing. So these are protein biomarkers. And the biomarkers, of course, can be used not only for biological entities, but also for new chemistry. Similar as for lead discovery, you have to have a relatively broad setup for immunoassays if you want to support all these areas. My mobile phone. Yeah, that can be the case. That was a good idea. We offer radioimmunoassays, ELISAs, of course, Luminex technology, which actually can be applied nearly only for biomarker assessments. So you have a lot of limitations when you want to use Luminex, for instance, for immunogenicity PK within clinical trials. We have mesoscale in-house, 2 machines in the US, 2 machines in the UK. We are highly using BioCore. I think we are relatively unique there. There are not many CROs where you will find this set up together. We're employing flow cytometry, immunophenotyping to support customers. We recently acquired a GyroLab instrument, so this is a most perhaps popular one at the moment for miniaturizing immunoassays. Most of our customers nowadays have a gyro lab in-house, and we offer particular cell-based assays to support, for instance, immunogenicity, so ADCC assays, for example. What we support is pharmacokinetic studies, and again, so this is for large molecules, so you know what PK is, so this is measuring post-administrative samples after administration of the drug, and simply look for the levels. And why is this done? So our customers want to know whether the drug concentration is above an effective level, is a concentration below a toxic level, and this, as mentioned before, can be highly variable, of course, depending on the matrix you're using, on the patient, on the how the patient is responding to the drug, and of course how the sample is collected. So for instance, when we're working on PBMC cells, we supply the clinical sites with a kit and a protocol exactly how to prepare their PBMCs, store them and send them as a batch to us. And we have somebody going out training those clinical sites for doing this, because this is not a trivial event. And of course, the absorption, elimination, and all the factors which might play a role in the response and the PK characteristics of the compound. Immunogenicity is something which has to be assessed for all antibody therapeutics. So it's a normal body's response to the drug. So it can be desired. For instance, if you have vaccine development, then this is exactly what you want. But it can be undesired, for instance, if the patient are developing anti-drug antibodies which can cause allergic or anaphylactic reactions. They can, of course, reduce the efficacy of your therapeutic, and they can induce autoimmunity, which is also quite nasty. And this cannot be only influenced, of course, by the drug itself, but also by the production process, formulation, and so on and so forth. So these are not trivial assays to set up, and customers working with us as a CRO, of course, have to trust in our competence and expertise to do those kind of studies, and there it helps a lot that we have a team like the team around Jeff Hale, who has really an experience in doing exactly this, bringing antibodies to the market. I think I already mentioned part of it. So why immunogenicity is done, or assays are done. So it's to find out whether the patient's generating antibodies, and if they do, whether these are specific ones, are there neutralizing ones? If there is a response, how strong is it? What is the titer? So what is the length or term of the response? Is it something which shows up only transiently? What class of antibodies are involved? So you have a different antibody answer, of course. It's not always the same. And of course the observed response, the result of disease, dose, route of administration, and so on and so forth. So these are the specialized services we offer mainly for monoclonal antibodies. Coming to protein biomarkers in drug development, I think this is clear to everyone in the room. Biomarkers are highly important. There are many companies which will not start a new program without biomarkers associated with it. So they play a role nearly everywhere. We talked about patient stratification before. That can be surrogate endpoints, can be companion diagnostics, but people might use biomarkers very early on for disease processes or detecting disease processes, for target identification, so on and so forth. So biomarkers can mean a lot of things to many people. They can be used in many different forms, and as a CRO, if we say we are offering biomarker services and solutions, then we have to react and find our niche within this plot and this major stream here. And what we think is, so first of all, there are two major differences whether you want to use a biomarker in a regulated environment or non-regulated. Non-regulated is very simple, and this is something you can get from a lot of companies, also from us. So this is data, biomarker data, only for R&D purposes, information purposes. Normally you use a kit as it is provided, according to kit manufacturer or manufacturer proposals, run it, report back the data, and that's it. Regulated is of course completely different, so it has to be done under GLP, GCP. Has to be specified in preclinical protocols and so on and so forth. So this part is completely different, and for a CRO is also involving much more consultancy for customers than doing a non-regulated way. And I think this was also mentioned on your last slide, fit-for-purpose models. So what is a fit-for-purpose model here? So you do not need the same kind of validation, whether it's analytical validation or method validation or clinical validation for all biomarkers like you would need it for a companion diagnostic, for instance, also, or to support a label claim of a drug. So there are different flavors in which represent more or less the different biomarker status on the drug discovery value chain I had before. So what we try to find out together with a customer who comes with us with a biomarker idea and wants to measure a particular biomarker, where on this plot his program and his needs fit. And according to that, we come up with an analytical plan how to do the validation. And unfortunately, we, not ourselves, so within our department, Merck could do that, but we can't do that, the clinical validation themselves. So we play more or less in this part to which extent we have to drive the analytical validation. And this is also then, for instance, limiting us from using Luminex, for instance, for later stages. It's nearly impossible with the matrix problems you get. So this is something where high consultancy is necessary. And this is where we see our major influence also, because we have people like Ron Bauscher in the US, who is heavily driving fit-for-purpose models for how to use protein biomarkers for clinical development. And with that, I would like to finish, and thank you very much. And I hope it was at least a bit enjoying, although it was not Theranostics.
So the session is now open for questions. So are there any burning questions for either Darren or Martin? Question on the end here.
It's a question for Darren. I was curious to know whether you could predict what the number would be on your first slide in the next 5 or 10 years. Does the $802 million go up, down, or stay the same in the theranostic development?
Would be a brave man who said it was going to go down, wouldn't it? But I think that is the, I mean, we were talking previously, and that is the key question, is that the drug industry has grown up with this high return sitting at the end of the day, and the processes within the industry and without, in terms of regulations, etc., are all built on that model, and it really is up to us and people in this room and all the people I mentioned in that slide set to build a new model where we can, you know, we've got great hypotheses, we've got molecules to prosecute those hypotheses, we've got to make it work from a financial point of view, whether it's AstraZeneca's investment board or whether it's a VC's. We've got to show that we can take that science and prosecute in the clinic and give them a return on their money. And just doing it the old way, we know it's not going to work.
Can I just have a follow-up in terms of, do you think the regulatory authorities will, given they're looking for stratified medicine, therefore cheaper health economics, push the decision-making earlier for the likes of an AstraZeneca to go down the stratified route so you lose the broader label opportunity, which was the top left portion of your graph?
So I might have misunderstood the question. I mean, I don't think the regulatory authorities per se are actually necessarily interested in the cost part of it. They are there for safety and efficacy, obviously. I don't think it's really necessarily a push or a pull. I mean, we all want to go there. That's where the science leads us, and that's what we should be doing. It's the right thing, and quite frankly, just developing a drug that we know from all our preclinical work and all the great science that's done in academia is predicated on a specific hypothesis, and then just chucking it into this heterogeneous disease that we've defined on anatomy and expecting us to get a return on that is ridiculous.
I think more of a Celldex example, where ODAC pushed them back and said you need a diagnostic. So if that's going to happen more and more, and the FDA are going to suggest that's the route, then you're going to make an early decision that I'm not going to go for the heterogeneous population.
Yeah, I think that's why you've got to say that's the probability of success. This is where the data are leading us. We need to engage, I think as someone said earlier, engage early and often and don't have these nasty surprises towards the end.
Thank you.
Roland.
Yeah, hi. You mentioned obviously the interesting concept that a lot of biomarkers really relates to both those speakers are generic. So in cancer, many companies are making drugs against the same targets, be it PI3K, be it HSP90, be it whatever. Therefore, there is a strong rationale for companies to work together, as you suggested, would be a good idea, to develop biomarkers collectively, because there's no commercial advantage necessarily in that. So what initiatives are with Millipore or with other companies to say, let's work together to develop an HSP90 biomarker that then not only helps you, but then because it's generically accepted, actually becomes accepted potentially by regulators downstream as well. So are there these initiatives in place at the moment, or are they being created, or are there EU initiatives in these?
Do you want to start now? Yeah.
Sure. So there are, of course. I mean, in the EU, there's this thing called the IMI, Innovative Medicines Initiative, and there are, and I think Martin touched on it as well, is that biomarkers can be all things to all people, and I would say that that there is definitely more activity in what perhaps is a pre-competitive space in terms of proof of mechanism. Proof of principal biomarkers that are used in the early decision making, and there's, I think it's been an easier place to set things up in the pre-competitive space. In terms of these predictive markers, as you can see from the presentation, it's pivotal to the product, and I think it's fair to say that we might have talked about things like the surrounding infrastructure, like the types of sample collection is a project in the US. In terms of that specific part that is so central at such a late stage to the drug, I find it quite hard actually to see how in the later stages that would work. But certainly for the early part of the development, I think it's a great plan. But I don't think we've got there yet. I don't know if.
No, I think I'd endorse your view. I think in terms of the earlier phases, certainly there is the FPR initiative in terms of the pre-competitive space, and certainly the UK has been trying to take a lead in that, and ABPI has been running workshops trying to bring companies together to absolutely facilitate the point that you make. But obviously in addition to that, there's the government funding of the translational research partnerships, which at the moment are in just one therapy cluster. But the idea is if they show feasibility and the pilot is successful, that they will look at other therapy areas where there's unmet medical need. And indeed, ideas like biomarkers and working together is some of the things that we hope to achieve from that.
It just seems to me that you're going to get by, the net gain by working together as a consortium around specific targets will be enormous relative to each one trying to do this alone, around the same pathways, basically.
Absolutely, and I've just come back from ASCO, I guess the largest oncology meeting, and MD Anderson were up there talking about the WIN consortium and other groups that are absolutely trying to do this. So almost we need to be tackling this on a global basis. Yeah. Any other questions, or are people kind of dying for lunch? Oh, one more question. Yeah, okay.
So how do you think industry and the regulators are going to cope with, if you accept that you're going to select the patient population, the next stage on is having to select the dosing cycle and the dosing frequency, and how are the industry and the regulators going to cope with products that have to be personalised to that level?
Megan.
Do you want to start now?
Well, I mean, I think to some extent we already have that in terms of there's a pragmatic approach to that. Certainly in oncology, it's a pragmatic approach to dosing, etc. And I think that's one of the key things about what gives a predictive biomarker legs. And if it's information that you can find out relatively quickly and safely, of course, to modulate dose, etc., then it's not a place where you would need to bring those kind of biomarkers in there. In a way, I mean, even the example I gave is actually about dose anyway. The gefitinib example is actually about the therapeutically achievable dose and the differential in hitting the target between ordinary tissues and the tumour. So it is really already about dose. In terms of taking it down to an individual patient, which is, I guess, one of the reasons why I don't particularly like the term personalised medicine, because really, in the drug industry, we're in the business of stratifying populations rather than personalising them. I mean, we're not going to do an N of 1, so it's going to be a question, if I understand correctly, there may be algorithms of what to do if, etc., but it's not going to be personalised per se for new drugs on that basis.
No, I'd agree. I think it's subpopulations we're looking at. Any other?
No, that's not my domain.
Okay. If that's it, close the session. Thank you for your attention. I think we're meant to be due back here at 2 o'clock. Lunch will be served in the Riverside restaurant, and then tea and coffee are actually going to be in the exhibition suite just outside. And could I ask you all, while you're enjoying your tea and coffee, as well as your networking, could you all please try and find some time to actually go and speak to the exhibitors who are here today? And thank you very much indeed. Thank you.